Okay. Is it visible now?
That's perfect.
Just a moment. Hello, everyone. My name is Sayantika Banerjee, and I am currently a fourth-year PhD student at Charles University, Prague, Czech Republic, and I'm also associated with the Institute of Botany, Czech Academy of Sciences. And today I'm going to present a little bit from the second chapter of my thesis, which focuses on how the effects of climatic factors such as temperature and moisture impact the root metabolites present in Rhododendron anthopogon. So I will start by introducing my study species, which I mentioned before, which is Rhododendron anthopogon, belonging to the family Ericaceae, and it is a high altitude shrub species found across the Himalayas. It forms dominant vegetation on sunny slopes and open patches. And it is a broad-leaved evergreen species with a conservative resource use strategy. Here are some of the photographs from my study areas, and you can see the patches of rhododendron in all these sites, which are a little bit brownish in color. With this, I come to a small background of what I'm doing on root metabolites. So what do we mean by root metabolites? Root metabolites means the chemical compounds which are present within the roots, which are produced within the roots. And it includes both primary and secondary metabolites. Primary metabolites include sugars, amino acids, etc., whereas examples of secondary metabolites are flavonoids, coumarins, phenolics, and other compounds. These metabolites are affected both by temperature and moisture. These are important to study because they have a very important role in plant growth, development, and defense and stress mechanisms. So with this little background, I will come to my, okay, here are some examples where people have done work with focusing on how climatic factors affect the plant metabolites. For my study, I am focusing on root metabolites, that is the chemical compounds which are present inside the roots. With this, I come to my specific research questions. So my first question is, how do root metabolomic profiles vary along natural climatic gradient, and do the effects of temperature and moisture occur individually or they interact in affecting metabolic profiles? And my hypothesis is that root metabolomic profiles are driven by both individual and interactive effects of temperature and moisture. Given that temperature is the primary limiting factor affecting plants in alpine regions, therefore the effect will be stronger. That is my expectation. The second question is that what specific metabolites and associated metabolomic processes are affected by climate and how? And my hypothesis is that climatic factors, through their interactive effects, will induce directional shifts in specific metabolites, and defense-rated metabolites will be present from the roots from warmer and moisture sites, and a higher number of secondary metabolites will be present as compared to primary metabolites. The third question is, how does soil chemistry affect root metabolomic profiles, and does controlling for soil chemistry affect the conclusion on the effects of climate? And my hypothesis is that climate affects plants both directly and indirectly via changes in soil chemistry. So there might be some effects of soil chemistry on plant metabolomic profiles. With this research questions, I come to my methods. So I conducted my study in the two Himalayan states in India, which is Himachal Pradesh and Jammu and Kashmir. And here is my study area. And the elevation of my study area varied between 3,200 and 4,200 meters. The mean annual precipitation varied between 400 millimeters and 1,200 millimeters. And the mean annual temperature during the growing season varied between 4 degrees and 10 degrees. So I selected 5 sites with different precipitation, and at each site I selected 3 elevations. So in total I had 15 populations. At each population, I laid 10-meter square plots, and from each plot, I selected 3 rhododendron individuals from which I collected the fine roots, stored them in silica gel. For the climatic variables, we installed these data loggers at all the 15 studied populations, which collected data for a period of 1 year and within an interval of 15 minutes every day. After bringing back the samples from India to Czech Republic, I followed this protocol and did untargeted LCMS to get the metabolite peaks. We got the data in 2 modes, that is positive and negative mode. With this, I come to and I used MetaboAnalyst and MS-Dial, these 2 softwares, to analyze the raw data and for other results, and I used R for doing the statistical analysis. Coming to the results, so my first question was whether the effects of climate occur individually or they interact. And in the positive ionization mode, we see that the effect of moisture was the strongest, followed by interactive effects. In the negative ionization mode, also the patterns were similar. And so we can say that moisture effects were stronger, and this was followed by the effects of temperature and moisture interacting, and in both positive and negative ionization mode. Here is the RDA plot, and we see the differentiation in the metabolomic profiles amongst the 15 populations, and that almost 50% variation is being explained by the environmental variables. And we can also see that how the interaction effect is also exerting important effect and important and significant effects. To see how many metabolites are affected by temperature, moisture, and interaction, we did this Venn diagram. And from this, we can see that 568 metabolites were affected by temperature, and 281 by moisture, and 496 by interaction in the positive ionization mode. And in the negative ionization mode, a total of 78 metabolites were affected by temperature, 44 by moisture, and 73 by interaction. So from this, again, we highlight the fact that interaction effects are important, and that only metabolites which were significantly affected only by a unique effect, only by temperature or moisture, is very less, and most of them are interacting with each other. After this, we tried to show the number of metabolites in each major metabolite class that were affected by the temperature, moisture, and their interaction. And we see that in the positive ionization mode, flavonoids were most affected, followed by amino acids, coumarins, and phenylpropanoids. And in the negative ionization mode, glycosides were most affected, followed by terpenoids, isoflavonoids, and lipids. Here we also see that the interactive effect of the effect of interaction of temperature and moisture on the metabolites present in the flavonoid classes were also significantly affected. All these metabolites that I have listed here, the metabolite classes mainly, they play some role in defense and stress, indicating that yes, temperature and moisture changes are affecting the defense-related and stress-related metabolites. After this, we did some pathway analysis to focus on, to get to know which are the main plant pathways that are being affected by these metabolites. And we found 23 major metabolite pathways. We used the MetaboAnalyst 6.0 version to do the pathway analysis. And here the bubbles represent the color of the bubble represents the strength of significance, and the size represents the strength. And here we can see that a total of 23 pathways were affected. I selected these few pathways which are very important from climatic stress point of view, and selected these metabolites. In the next slide, I will show that how the direction changes in the production of these metabolites in response to climatic factors. And we see that high temperature and high moisture is leading to an increase in ferulic acid, syringin, and pantothenic acid, which play important role in stress mechanisms, and the opposite is observed in the other two metabolites. After this, we did a network analysis, and we can see that both in the positive ionization mode and negative ionization mode, there are a lot of metabolites which are acting as hub metabolites, that is the bubbles. Here, the edges represent the strength, and the color, green color, represents the positive correlation, red represents the negative correlation, and the thickness represents the significance and the strength in between these metabolites. The main thing to observe in this correlation network analysis is that they are all present in a very integrated manner, showing that changes in one will affect the changes in the others. From this network analysis, I selected a few important ones. And here we see that flavonoids, phenolics, and glycosides, which all of these three, which play important role in defense and stress related mechanisms, increases with increase in temperature and high moisture levels. And the opposite is observed for alkaloids and amino acids, which fall in the primary metabolite classes. So to conclude, the other third question, where I want to see whether the effects of soil chemistry is significant or not on root metabolite. And in the positive ionization mode, when climate is used as a covariate, soil electrical conductivity has a significant presence, and we see the same in soil in the negative ionization mode. And in the negative ionization mode, when we do not use climate as a covariate, the effect of soil nitrogen and soil pH is also significant. So this shows that soil chemistry does affect the root metabolomic profile. So to conclude, I come to my first question: how do root metabolomic profiles vary along climatic natural gradient, and do the effects of temperature and moisture occur individually, or they interact in affecting metabolic profiles? And my first answer is that moisture effects were stronger. Significant interaction of temperature and moisture were seen when soil chemistry is used as covariate. So the hypothesis we had that soil chemistry might affect the impact of climate is not true. And we see that the temperature effects were not significant individually, but in combination with moisture, the interaction effects were significant. The second question, that what specific metabolites and associated metabolic processes are affected by climate and how, we see that both primary and secondary metabolites were affected by climate and its interaction. Most of the metabolites that were affected by climate and interaction belong to the defense related metabolite classes. A total of 23 metabolomic pathways were significantly affected by climate and its interaction. And a highly connected metabolome in the network analysis suggests that the pathways operate in an integrated manner. The third question, that is, how does soil chemistry affect root metabolic profiles? We see that soil chemistry does affect metabolic profiles, although the effects of climate do not change due to soil chemistry, and the effect of soil electric and conductivity was the most significant. With this, I come to the end. I would like to thank my supervisor, Zuzana Munzbergova, for her help and her with the analysis. I would also like to thank my advisor, Dinesh, and my collaborators. I would like to thank my university, Charles University, the Institute of Botany, and the Institute of Microbiology of the Czech Academy of Sciences for giving me the lab to work on my analysis. And here are my details if you want to connect with me or ask me any further questions. Thank you so much for your time.
Great talk. Thank you. So we have plenty of time for questions. The way you're going to ask your question is either you can type it in the Q&A, or you can raise your hand. And there's only a few of us here, so if you raise your hand, I will give you the talk permission. Martin.
Hi. Um,
Hello.
did you compare your studies with plants and culture if this has the same effect?
Sorry, plants of what?
Did you compare your studies with plants and culture?
I use natural populations.
So you don't compare them with the plants and culture nursery produced?
No, not in culture. I use natural populations. I collected the samples from I had 15 populations across 5 sites, and I collected plant samples from the natural populations. I did not focus on plant cultures.
Okay, we have anthopogon in our garden and culture in the nursery fields,
Yeah.
and we have two different selections of it.
Okay.
One with heavy endomentum and the Betty Graham cultivar. And Betty Graham is much easier during hot summer days as a heavy endomentum one. And both need more moisture during culture in the summer months, and they don't tolerate dry substrates. Did you make the same experience observations in the field?
No, not exactly, because we collected the samples within a span of 2-3 months when the window is open to do the work there in the Himalayan region. So we did not focus on this. We just collected the samples, and our main question was to see how temperature and moisture is affecting this. And
Mhm.
from the data loggers, we used the growing season temperature and the soil water content present in the soil during the growing season for doing the rest of the analysis, because my focus was only how to see the effect of climatic factors.
Okay,
Thank
thanks.
you. Thank you for your
Martin,
question.
we have a talk coming up in a minute, which I think might be speaking to your question.
Okay,
I
thank you.
think the next talk might answer your questions a little bit more about that.
Okay.
My thoughts were similar in terms of thinking about how the results you have impact the things that people like about rhododendron anthopogon. One of them being that it's a nice plant to grow. It's a very attractive plant. And then the other being its medicinal chemistry properties. So based on your work, it looks like someone who was going to grow the plant in culture might be able to think about how they could manipulate the conditions that they're exposing it to in order to alter the metabolic profile. Do you think that's true?
Yeah, that will help. Like my entire study, this is just the second chapter where I'm focusing on specifically the metabolites, and I showed the metabolite names. But the metabolites that are present will help in recruiting several fungal and root other bacterial biota, which is the objective of my other chapters. And with the biota recruitment, it will help us in understanding whether how good the plant is growing in moist, warm conditions, or under, like in certain sites which I have are very cold and less moisture, that is stressful. So the studies with biota will help in making the choices, how to grow better the rhododendron anthropogon, I think so.
Ooh, I'm excited for those results. There's been, yesterday there were a couple of talks on
Yeah,
rhododendron biota, and I
yeah,
think just thinking about how they recruit and what the signals are, that's very exciting.
yeah.
Wonderful. If there's no other questions, thank you so much.
Thank you, everyone.
Yes, and I think we'll move on to our next talk, Dinesh Thakur, also from the Czech Academy of Sciences, Growth Dynamics of Himalayan Alpine Shrub, Rhododendron Anthropogon Across Climatic Gradients. Hi, Dinesh.
Hi.
Go ahead and share your screen.